let's talk about some of the details having to do with photosynthesis. So first of all, the basic way that we're going to do the light reactions or the light dependent reactions is we're going to use sunlight energy, and sunlight energy is going to excite electrons that are part of chlorophyll, the chlorophyll molecule. And that excited electron, if I were to just excite it and let it just fall back down, it would either emit heat or light or both. But instead, what I'm going to do is control the fall of the electron, just like we did before in the electron transport chain, and allow it to make, hold on a second, I'm looking for the right one, allow it to make ATP like it did in cellular respiration, and also NADPH, which is an electron carrier. So just kind of a brief overview is I'm going to have chlorophyll that is found in what we call photosystems. Talk a little more about that in a second. I'm going to excite electrons with light. I'm going to let them fall back down in a controlled way through an electron transport chain and make ATP, just like we did in cellular respiration. And then the second time it gets excited, I'm going to collect those excited electrons and use those as an energy source. And we're going to do the whole thing, that is, as I showed on the last video, we're going to use this ATP and NADPH in the second part of the process to make sugar. Okay, so let's just talk about the photosystems a bit more. First of all, let's just talk about where they are. Okay, so this is a depiction of the chloroplasts and the grana that are inside the chloroplasts, and they are covered with thylakoid membrane. Thylakoid membranes will have these photosystems embedded in them. That is, the pigments will be embedded within the membrane. And this is a depiction of chlorophyll A or B. They both have, in the middle, they have a magnesium, that is an ion, and that ion can take on the electron and or, if it's excited, give it off. So that's going to be kind of like the central part of the molecule that is taking the electrons and giving them away. Okay, so looking at the photosystem a little bit closer. So each one of these things that we call photosystems, they are a whole bunch of different pigment molecules. Now they can include any of the four pigments that we talked about before. That is chlorophyll A, B, and the carotenoids, carotene and xanthophylls. Those are all going to funnel the energy, and this is kind of like a satellite dish, where it's going to funnel the energy to a central point, or a central, or what they call a primary electron acceptor. So this, I'm sorry, I said that wrong. They're going to go to the reaction center chlorophyll. So the reaction center chlorophyll is where all of the energy from all of the molecules that are collecting light energy are funneled. Now that reaction center chlorophyll is the one that is going to give off its electron. That is, the electron is going to be excited to a higher energy state, and it will be collected by the primary electron acceptor. All right, so this part, the electron acceptor and the reaction center chlorophyll, are both called the reaction center. Okay, so these other molecules are all what they call the antenna pigment molecules. And in essence, like I was saying, it acts like a satellite dish, and it's going to funnel all that energy there. But light is doing that. Now they just show light hitting one to show you how it might be transferred, but literally it's hitting the whole thing at one time. So all of them are transferring energy to that reaction center chlorophyll that's then going to excite the electrons. All right, so the basic process goes like this. Okay, I'm going to back up here, and if you want to, you can take notes on this part, so you could stop and take notes. First of all, let's just say we were talking about the chloroplast, and some specific parts of the chloroplast that we want to consider are the stroma, which is in the internal structure, that is the space inside. It's kind of like the matrix in the mitochondria. And then we have these stacks of grana that have thylakoid membranes on the outside. So these are the little loops. And inside of that is called the thylakoid space. Okay? So what we have depicted here is we have one of these thylakoid membranes on a single grana or single stack. And you have the stroma side on this side and the thylakoid space on this side, okay? And what is happening is you have light that is going to hit that photosystem, and that photosystem, the chlorophyll, or the reaction center chlorophyll, will have that electron excited to the primary electron acceptor, okay? So that's what's going on right there. All right, now this, the electron then is gonna fall to the various proteins, and just like we talked about before, each one of these electron acceptors is a slightly lower energy level than the previous one. So the electron will go from that one to that one. Now as that electron gets taken on, it also takes on a hydrogen, and as it flows down to the next one, it dumps the hydrogen in the thylakoid space. Likewise, this one takes on the electron, takes on a hydrogen, and as it flows down, it dumps the hydrogen into the thylakoid space. And it keeps doing that all the way down to where it is collected by the next photosystem. All right? Then what happens is those hydrogens will flow through an ATP synthase, and they will make ATP, just like they did in cellular respiration. So if you don't remember how that goes, go back and look at that video again, okay? Now the second part is that the electron from this part will be collected by this photosystem. It gets excited also by sunlight, and then the excited electrons will be collected in the form of NADPH. So this is, in this process, one electron then can go from here to there, to there, to there, be collected off in its excited state, and meanwhile making ATP. All right, now the amount of ATP and NADPH are equal in what this in this part that we were talking about, which is called non-cyclic electron flow. That is, the electron is not going around in a circle, it's just going in one direction. Okay, now some details about this I will show you that I haven't mentioned. Okay, one thing is that, first of all, that water is going to be donating the electrons. And as water donates the electrons, what is produced is oxygen gas, all right, plus hydrogens. All right, so what's going to happen there? So this is the part of the process that produces oxygen. Okay, as those electrons then get excited, we're going to make ATP, as I said before. Now the second thing that I haven't talked about is this real confusing thing, which is this one is called photosystems 1, and this one is called photosystems 2. Now that's confusing because photosystems 1 in this non-cyclic flow is coming first, so to speak. That is first in the sense that the electron enters here and ends up at the end out of photosystems 1. Now the reason why they are called this is because photosystems 1 was discovered first. And then later on, photosystems 2 was discovered, and then various things about the process. This process has been worked on for a long time, and they, you know, have many different people who have worked through the process trying to figure out what's going on. Okay, so this is what we call non-cyclic electron flow. That is implying that there is something called cyclic electron flow. Okay. Before we go on to that, though, let me just show you another depiction of this so that you can see how the ATP is perhaps being made. Oops. Okay, so this is the stroma, and then this is the thylakoid space, and they're showing you the light and oxygen being produced, but then you have the flow of the electron, eventually going to NADPH, and the flow of the hydrogens now that have collected in the thylakoid space, making the ATP. The reason why I drew it this way, and why the other diagram drew it this way, was just to show you that this electron transport chain is the one that is pumping the hydrogens and making the ATP. That is the electron chain associated with photosystems too. Okay.